And we identified over 4,000 proteins that were in the chromosomes. And so, of course, most of the chromosome is the histones because they're in the nucleosomes, but they're not in the scaffold. We get rid of them. So what is in the scaffold, amongst other proteins, is a group of proteins called SMC proteins. So SMC proteins organize the behavior of mitotic chromosomes. So, for example, a complex of SMC proteins called condensin, that's what's responsible, I'll show you today, for shaping mitotic chromosomes. And another complex called cohesin is what holds the replicated DNA molecules together so that when the cell divides, each replicated DNA molecule splits so that one goes to each daughter cell. And this cohesin complex is also very important in organizing loops in interphase. And so mutations in cohesin proteins cause Cornelia de Lange syndrome. It's a developmental disorder and other disorders. So there are many people who study gene expression who study SMC proteins, but there are also cohesin, but there are also people who study chromosomes. Condensin 2 acts first. Condensin 2 is in the nucleus, and it acts first. Condensin 1 is in the cytoplasm, and it acts second. All right, so that top band was topoisomerase 2. That band turned out to be SMC, a protein called SMC2. And SMC2 is that protein. So there are two condensin complexes, but the way they work is that they both have SMC2 and SMC4. They have two different sets of these guys. So if we get rid of SMC2, there's no condensin, neither complex. They're both gone. So that's what we're going to do. We're going to get rid of SMC2, and the condensin complex is going to fall apart, and we're going to ask, what does this do to the structure of chromosomes? To a cell where we've gotten rid of the condensin, so there's no condensin. So the first thing we do is we section. And then using a very sophisticated image processing protein, we do what's called modeling. So that is, we recognize the chromatin away from the rest of the cell. And then we do segmentation, which is we separate the separate chromosomes. And then we can analyze it. But what you can see is this doesn't look like we got rid of condensin, and this doesn't look like mitotic chromosomes anymore. So a less fancy way of doing that is just to look in the light microscope, and on the top, we see cells entering mitosis. And here we see cells entering mitosis under the same time scale, but without condensin. And this looks like an interphase nucleus. It doesn't look like mitotic chromosomes, but look how big it is and how big that is. And remember that volume goes with the cube of the radius. So this is highly condensed. This is much smaller in volume than that. So the chromatin is highly condensed, but it does not form chromosomes. So you require condensin in order to form chromosomes. So now we'll talk about some of our work on chromosome formation. So what happens to the SMC proteins during mitotic entry? So we're gonna do this protocol where we're going to arrest the cells, we're going to release them into mitosis, and then at very short intervals afterwards, we're going to let them off, and we're going to ask, in the first case, so this is a marker that Kamiko Samajima made. So this is it's 4 times GFP hooked up to a nuclear export sequence. So this lives in the cytoplasm. And so there's the empty nucleus. And at this point is when the nuclear envelope breaks down. This is 3-minute time points going into mitosis. And because the synchrony we have with this experiments is so perfect, we know exactly when the cells went into mitosis. And so this is when the nuclear envelope starts to break down. But actually, if you look in the middle, you can see proteins from the cytoplasm are coming in before. And so here you can see that. So proteins from the cytoplasm are coming in before the nuclear envelope breaks down. That's a surprise to many people. But then if we look at the behavior of the molecules I've been talking about, condensin 1, well, remember, condensin 2, that's the one that lives in the nucleus. So it's there in the nucleus. And actually, over the course of mitosis, a little bit of it comes off of the chromosomes. Cohesin drops quite a lot. as cells go into mitosis. And everybody says that cohesin is gone in mitosis. And then condensin 1 is the protein that's in the cytoplasm, and it increases a lot. And this is quite important that it does this, and you'll see why in a minute. But this is by normalizing the amounts that we've detected in the mass spec to what's there in G2. What if we take the absolute numbers and score how much of these molecules is there? Then we get something that, when this paper is submitted, is going to be we're going to have a bad time with referees. Because there's a ton of this cohesin molecule. So even though the levels drop, they only drop down to about the level of the molecules that are supposed to be making the chromosomes. So there's still quite a lot of these cohesin molecules on mitotic chromosomes. So the first study, which I'm going to summarize for you in the beginning, we only studied condensin because only condensin is supposed to be involved in making chromosomes. In the last work I'll talk about, at the end of the talk, we asked about what does cohesin do? And cohesin does a lot. It's not supposed to, but it does. So now I'm going to go to our work that we did looking at high C and modeling. So at the beginning of mitosis, one of the things that we saw, or what we saw in the high C map, was this kind of not very interesting line. And this is called a scaling plot. And this is 10 kb out to 1 gigabase here. So it's a plot across the size range in the chromosome, and this shows how likely sequences are to be cross-linked to one another. And so if we took this plot and we analyze the data going away from that line, it's like analyzing the data going along on this line. And this means that for the features that we see here, we can give them an absolute size. We know how big they are because that's an absolute scale. And the only interpretation, the polymer interpretation of this structure that works, is that what's happened to the genome in the beginning of mitosis is just got turned into a chain of loops. So in the interphase nucleus, it was organized into heterochromatin and euchromatin. In mitosis, it's just a chain of loops. And in the beginning, these loops are, because we know the size, these loops are about 60,000 base pairs. But that size changes, and that's not important. Okay, so that's what we saw in the beginning. But we saw something later. What happened was nobody had ever seen anything like this in a high C map. You not only had the middle line, but you had these other lines that appeared on either side of it. And what do they mean? So the lines tell you that sequences are close together. What this map is telling me is that every sequence on the arm of this chromosome is close to sequences 10 million base pairs away. And here it's 3 or 4 million, here it's 5 million, here it's 10 million base pairs away. How can every sequence be close to sequences that are 10 million base pairs away? It's a helix. So you have the normal high C map you have, which is things that are close to each other. But if you go between the steps of the helix, then depending on how the helix spirals up, you can be up to 10 million base pairs away. So that told us that the chromatin, if you've heard of TADs, they're hidden in this line, and these are compartments which are active and inactive chromatin. And so what they saw, that what they had seen previously and studied, they had studied mitosis before in Job Decker's lab. They saw that the interface structure goes away. They saw that. But what they had never seen was that the second diagonal appeared. And the interpretation of it is that chromosomes are organized like a spiral staircase with a loop coming off of the end of every step. We started in prophase with loops which eventually become quite large, 400 kb. And then condensin 1 jumps on and it makes loops inside those loops. And then you end up with this compact structure. That was where we were in 2018. So we're still writing the paper because you try writing a paper with a bunch of theoretical physicists. Let me tell you, what happened? If there's cohesin there and the cohesin is holding the two replicated DNA molecules together and condensin is trying to make loops, how can condensin make loops if cohesin is there? So one possibility is that when condensin hits cohesin, it stops. I mean, that would make sense, right? And that would give you an organization like this. And Samir Abraham has just recently finished his PhD. Here he modeled what would happen if we have condensin hits cohesin and it stalls. So you would end up with a structure. This is two sister chromatids. But you would end up with a structure that looks like that. And we know that's not what things look like. So it turned out that the only way you can end up with what looks like chromosomes is that when cohesin hits cohesin, it can jump over it somehow and keep extruding the loop. And how you do that topologically, pushing the molecules to the ring, I don't know. It doesn't work like that. In fact, we don't know how it works. But Samir predicted that if that would be the case, his simulations show you can actually separate the two sister chromatids, and you end up with two sister chromatids separated and cohesin in the middle. So if you did a diagram of it, it would look like this is the blue is the DNA. This is the scaffold, the condensin in each arm, and this is the cohesin in the middle. If you actually look at cells and you're able to see all these molecules, which is extremely difficult to see the cohesin, you can see the condensin on either side and the cohesin in the middle. So this is data. That's a model. That's pretty good, I think. So, this is the helix with the spiral staircase in the middle. Let's get rid of condensin 2. That's the one that was acting in interphase and making the big loops. And so we have only the one that makes the nested loops, and they're smaller loops. So we have squiggly looking chromosomes, long and thin squiggly looking chromosomes. So if we get rid of condensin 1, which makes the nested loops, we should have only big loops. And so we have short, fat chromosomes. But now let's get rid of cohesin. They don't stay the same. They don't look the same. First of all, you can see we've gotten rid of cohesin because the sister chromatids are separated, but they look really weird. And so we decided to look at what these look like by high C. Okay, now let's get rid of the one that makes the little loops. And this high C profile, it's insane. There's a diagonal, there's another diagonal, and there's another diagonal. So these chromosomes have a very bizarre, it's like a helix of helix of helix. It doesn't make sense. And if you get rid of the one that if you get rid of condensin 2, you have no helix, no nothing. That looks like a super simple pattern. These look super complicated. Then the other thing Fernanda needed to know was what's the diameter of the chromosomes. That was another thing that Anton wanted to know. So using a Mira, she can define the midplane of each of these three-dimensional maps, and then measure the diameter. So those are the three different types of chromosomes. This is no condensin 1. So this is big loops, lots of tiny loops. And what you can see is that's not a cylinder. In fact, the problem that Anton had was he was doing his modeling and doing his modeling and doing his modeling, and his modeling all involved setting up a cylindrical chromosome and moving the polymer inside it. The only way you could model this was to set up a random coil. It's a random coil, so it's fundamentally different organization. Condensin 1, they're not nearly so nice as that. If you don't have condensin 2 is responsible for making the helix, and it's responsible for making the chromosome a cylinder instead of just a random coil. So we have long, floppy, random chromosomes. If we don't have condensin 1, the one that jumps on, we have these huge helical loops. The weird thing is the high C profiles look amazing, but the chromosomes look very messy.